program is sponsored by North Central SARE, and the project team includes Kansas State University faculty from plant pathology, agronomy, horticulture, and entomology. The entire program includes these introductory webinars, hands-on workshops, and optional follow-up online learning community activities to explore the topics in more detail. This webinar will focus on soil as a living system. My name is Megan Kennelly, and I'm an associate professor in the Department of Plant Pathology at Kansas State University. My position includes research, plant disease diagnosis, and outreach and extension for horticultural crops, including vegetables. For this presentation, I need to acknowledge Lorena Gomez and Karen Garrick. Lorena is a PhD student, and Karen was a faculty member here at KSU, and now she's at University of Florida. Lorena is researching microbes in vegetable cropping systems, and I have borrowed some slides from them. Another key resource in building this presentation is the SARE book, Building Soils for Better Crops. And we'll be using that book at the workshops a little bit and in the online learning community. So in this webinar, we'll be exploring different kinds of microbes. This includes bacteria, fungi, viruses, and nematodes. In the webinar and the workshops, you might be hearing the word microbiome. Microbiome is a term to include all the microorganisms associated with a system. You might have heard the term human microbiome. There's been a surge of recent research to explore the microbes in our digestive tract and other parts of our bodies. So you may have heard this term in the news. Another term is phytobiome. So phyto is for plant. So phytobiome is all the microbes associated with plants. And more specifically, the rhizobiome refers to the microbes in and near the roots of plants. The American Phytopathological Society is spearheading a new effort called the Phytobiome Initiative to help call attention to the microbiome of plants with connections to soil science, entomology, and other disciplines. In the webinar, I'll just briefly mention some larger organisms such as arthropods and also the plants themselves, and I'll talk about some of the methods we use to study microbes. So microbes are organisms that are mostly unicellular and typically too small to be seen with the naked eye. We'll be exploring some of these microbial groups, including viruses, bacteria, fungi, and nematodes. Some other organisms that I won't specifically discuss today would be algae and amoebas. So bacteria are single-celled organisms with a pretty simple structure. They inhabit soils in every ecosystem. And some bacteria can live in very extreme environments, like deep down in deep sea vents. Here is a microscopic image of a bacterial cell showing the kind of main oval-shaped cell body and some tail-like appendages. Here is a cartoon of a bacterial cell showing the main parts of the cell and that tail, again called the flagellum. So bacteria have a nucleus, a cell membrane, a cell wall, various other cell components, and basically everything they need is packed into each cell. Bacteria have many different functions. They play a crucial role in the decomposition of organic matter, and some bacteria are very good at breaking down complex molecules, and they can be players in remediating soils that are contaminated. Bacteria play roles in the overall carbon cycle, both fixing and releasing carbon. And there are actually tests of carbon dioxide fluxes in soil that can give us a picture of overall microbial activity in that soil system. Bacteria are also critical players in the nitrogen cycle. So nitrogen gas in the atmosphere is not a very active compound, but nitrogen is an important component of many biological molecules, including DNA and proteins. And bacteria are critical in this cycle. So, for example, specialized bacteria associated with nodules on the roots of legumes fix nitrogen gas to ammonium. There are some soil-dwelling bacteria not associated with those nodules that can also fix nitrogen. Other bacteria convert ammonium into nitrites, and some convert nitrites into nitrates, and some bacteria convert various nitrogen-containing molecules back into nitrogen gas. Here are some photos of the nitrogen-fixing bacteria that form associations with legumes. So the term mutualism describes a relationship between organisms that is mutually beneficial. With these nodules, the plant provides food and a source of carbon from doing photosynthesis or making sugars. So it provides that to the bacteria, and in turn, the bacteria fix nitrogen and supply it to the plant for building, proteins and DNA and all of those things. Okay, so now we'll switch and talk a little bit about fungi. So usually if you say fungi, the first thing that pops into people's minds are mushrooms, but there are many other types of fungi. So at the organism level, fungi are more complex than bacteria, and instead of just being one-celled like bacteria, most of them are multicellular, and they form more complicated structures. They are common in nearly all habitats. People used to think that fungi were related to plants, but they are actually more related to animals than to plants. So sometimes you'll see in old books that fungi are small plants lacking chlorophyll, but that is not true. They are more related to animals. Okay, fungi have many different functions in our ecosystem. They play roles in the decomposition of organic residue. They are important decomposers of lignin or wood. Some fungi are mycorrhizae, and they have a beneficial symbiosis or mutualism with plants. And here's an image of a root with mycorrhizae. Some fungi produce antibiotic or antifungal compounds, and people have developed products based on those. Probably the most famous would be penicillin, which was derived from the penicillium fungus. The strobilurine fungicides, a modern class of fungicides, was actually derived from a compound produced by this mushroom. So these fungi aren't producing these antibiotics or antifungals for us, like out in the environment. They are producing them to ward off competitors, but people have developed and used those compounds. Okay, I mentioned lignin breakdown. Fungi are critical in the breakdown of wood, and in most cases this is beneficial. Wood decomposition is important for recycling those nutrients. In the case of urban trees, sometimes wood decay can be a problem, of course, if you have a decaying tree hanging over your house or your deck or something. But overall, the breakdown of lignin and cellulose is a critical part of nutrient cycling. Okay, so viruses. Viruses are tiny. They are too small to see with a standard microscope. Viruses infect living cells, and they require a living host. They cannot survive on their own. Viruses infect all types of organisms, including humans, animals, plants, and even other microbes such as bacteria. The viruses are host-specific, though. So, for example, the viruses that infect people and cause flu, for example, do not cause disease in plants, and plant viruses do not cause disease in humans. Here are some images using an electron microscope showing some viruses. So here we see kind of outlined. We've got a Gemini virus on the cassava plant, and on the right is an electron microscope image of the influenza virus. Here we've got a tomato infected by a virus, and this picture shows a virus that is actually infecting a bacteria. So as I said, microbes can infect other microbes. All right, so nematodes are animals. They are tiny microscopic worms, so maybe they don't really count as microbes, but I'm just going to go ahead and include them here. Soil-dwelling nematodes live in water films surrounding soil aggregate. So they don't live in 100% dry environments. They do require that water film. Some nematodes are plant parasites, and there'll be a separate webinar all about plant diseases. So here's just a few points here. So plant parasitic nematodes are fairly small, from about 0.5 millimeter to 2 millimeters in length. Here's an image of a type of nematode called ring nematodes. This photo shows us a section of root, and those long skinny things inside are the juveniles of the root knot nematode. So those are colonizing the root and causing damage there. All of the plant parasitic nematodes have a needle-like mouth part called a stylid, and that's what they use to kind of puncture into plant tissue and acquire nutrients. So those are the plant pathogens, but most nematodes are beneficial. Some of them break down organic residue and are involved in nutrient cycling. Some nematodes infect insects. Actually, in greenhouse production, like in the production of greenhouse ornamentals, you can actually purchase nematodes that are used as biological controls of some insect pests. So they can rear them in mass numbers and ship them out to growers, who then release them in their greenhouses as a beneficial organism. This image shows the kind of tip of a bacterial feeding nematode. So this one eats bacteria, and you'll see that it does not have a stylet like those plant feeders would have. Okay, so now we've talked about some different kinds of microbes. Some of them are plant pathogens, and again, there will be a separate webinar about some vegetable diseases. The plant pathogens can reduce plant growth, affecting both yield and crop quality. And there can also be microbes that are human pathogens on plants, and occasionally we find, hear stories about that in the news. But just remember that most microbes are beneficial. Turning now from microbes, we'll talk just a little bit about some of the larger soil-dwelling organisms. So one would be earthworms, and they feed on plant residue in the soil and organic matter. They have an important function mixing soils. So earthworms can move 1 to up to 100 tons of soil per acre per year, and they are referred to as nature's plow in that SARE soils book I referenced before. So they're important contributors to nutrient cycling. Earthworm burrows can also facilitate rain infiltration as another benefit. Insects and arthropods are numerous in soil environments, and Dr. Raymond Cloyd from entomology will be the lead on talking about insects and their interactions with cover crops and lots of other topics. But here's just a few points to lead off here. So some insects eat other insects. Some insects eat seeds down in the soil. Millipedes, which are not insects, but they're arthropods. They consume plant residue. And centipedes are predators. All right, so when we're talking about the living soil, we need to talk about plants too. So with plants, sometimes we kind of forget about the underground part and only think about the above ground part. But plant roots have complex interactions with other organisms in the soil. Small roots help soil bind together and form aggregates. Plants are leaky, and they exude chemicals that are food for microbes. And the plant, the environment near the roots is called the rhizosphere, and the microbes there in that area are called the rhizobiome. I used that term before. So again, microbiome is a generic term for all the microbes associated in the environment. Phytobiome is the microbes associated with plants, and then even more specifically, Rhizobiome is more specific to the microbes associated with the root. Okay, so what's biodiversity? Biodiversity is a term to describe the different organisms in an environment, and it can be measured in different ways. So one way to look at biodiversity is to kind of define and count which species are present. Another way to look at diversity is to consider the functions of those groups. So for example, some organisms are doing photosynthesis, others are decomposing organic matter, some are fixing nitrogen, some are producing antibiotic compounds, some are predators. So those functional groups and what they're doing in that environment is another measure of diversity. And soil management practices, or cover crop practices, can have major effects on biodiversity. There's a lot we do not know about microbes. And here is one quote. It is a common misconception among biologists and non-biologists that bacteria are relatively well known because they are so important in medicine, ecology, and molecular genetics. But the truth is that the vast majority of bacterial types remain completely unknown, with no name and no hints of the means needed to detect them. So that's a quote from E.O. Wilson back in 1992. All right, so how can we study these microbes if they're so difficult to work with at times? Traditionally, methods for identification of microbes involved culturing on Petri plates. So this provides a mechanism to basically get them into captivity, to study their growth, and to identify them and do various experiments to learn some things about their function. For example, you can grow them on media with different types of nutrient sources to learn about their metabolism. That is, what do they eat and what types of compounds do they produce. You can combine them in different ways or put them on plants to see if they're pathogens. You can do all sorts of stuff if you can get it into culture first. So there are, again, many advantages of this type of technique. It's useful for understanding various growth characteristics. You can study their function. And these studies have revealed a lot of information about the diversity of these microbes. But there are disadvantages too. And a really important one is that many microbes just do not grow in culture, or we just have not figured out yet how to grow them. It's been estimated that less than 0.1% of microbes in soil are culturable. So because of those limits, over the past 20 years or so, the methods for identifying soil microorganisms have changed dramatically, giving us a much better picture of diversity, but there's still a lot to learn. So with these new approaches, there's ways of studying microbes that do not rely on culturing them, and we call these culture-independent methods. So this provides a big opportunity to gain information about microbes that we were not able to study before. These methods are critical to studies of the overall microbiome. So these new DNA-based techniques estimate that the number of bacterial species in the soil is 200 times higher than our estimates from culture-based techniques. And this was revolutionary. All that was known before was based on that less than 0.1% of bacteria that we could culture. And similar estimates have been confirmed for soil fungi. So another way to think about this is that these newer DNA-based techniques give access to an estimated 90 to 90% of the unculturable soil microbes. So let's back up and remind ourselves about DNA and RNA. So DNA and RNA are significant components of all living organisms. They're present in all microbes, and they carry genetic information as the code of life. DNA and RNA are composed of long chains of molecules called nucleic acids, and the sequence of those molecules can provide us information about the identity of the organism. These nucleic acids provide the code for building proteins as well. So once we know about proteins, we can understand more about the function of the organism. So how this works in soil is that you basically collect soil and you extract all the DNA from it, and then you can sequence that DNA, and from that sequence information, you can figure out basically who is there, which microbes are there, and some indication of their function, that is, what are they doing. So soil is a complex living system with a lot of players, and this webinar introduced you to some of those players. We'll be covering some of these topics in more detail at the workshops and in the follow-up optional online learning community. So here's that outline again. We touched on the different kinds of microbes and some terms associated with microbes like microbiome and rhizobiome. There are other players in the soil, like insects and other arthropods. Plants have a significant influence on the microbes, and we talked a little bit about how researchers study microbes. So that brings us to the end of this webinar, and we'll be exploring other topics and other webinars in those workshops and the optional online